Hello, I'm Kachax. Let me explain about the stages of the zygote formation. The first stages is capacitation. In these stages, I remove my glycoprotein coat and rearrangement of intermembrane particles occur. The second stage is acrosomal reaction. The hydrolytic enzymes released from the acrosome make a hole in the jelly coat. The acrosomal process protrudes from the sperm head, penetrates the jelly coat and binds to the receptors in the egg plasma membrane. The third stage is cortical reaction. Fusion of egg and sperm plasma membranes triggers release of Ca2+ from the egg's endoplasmic reticulum into cytosol. High concentrations of Ca2+ cause cortical granules to fuse with the plasma membrane and release their contents into zona pellucida. Enzymes from granules thicken and harden the zona pellucida. This resists entry of additional sperms. And the last stages is activation. High concentrations of Ca2+ in the egg will stimulate increase in rates of cellular respiration and protein synthesis. Cytoplasmic pH changes from acidic to alkaline due to extrusion of H+. Metabolism of activated egg increases, the sperm nucleus swells and merge with the egg nucleus forming diploid zygote.
This is morula. From zygote, it is undergo mitosis 5 or more times. It has 32 cells or more. At this stage, morula does not have blastosol. This is blastula. Blastula is further development of morula by more mitosis. It has 64 or more cells. At this stage, it has blastosol. The outer unit layer around blastula is called tropoblast. The mass of cell inside blastula is called inner cell mass.
At this stage, the embryo is called a blastocyst, the mammalian version of blastula. The clustered one end at the blastocyst cavity is a group of cells called inner cell mass, which then develop into embryo. Implantation of the embryo is initiated by the tropoblast, the outer epithelium of the blastocyst. The enzyme secreted by the tropoblast will break down the molecule of endometrium and also extend finger-like projection. Around the time the embryo undergo implantation, the inner cell mass of the blastocyst will form a flat disc with an inner layer of cell, epiblast, and the outer hypoblast. Following implantation, the tropoblast continue to expand into the endometrium, and at this stage, gastrulation start.
By the end the second week of development, the biluminar embryonic disc consisting hypoblast and epiblast has formed. Throughout third week of development, this biluminar disc differentiate establish three primary germ layers. In the process known as gastrulation, around 15 days after fertilization, a freaking structure formed along the midline in the apoblast mirror of caudal and biluminar embryonic disc. This is called primitive streak. At this stage, the formation of primitive streak define the major body axis of the embryo, including the cranial end toward the head and caudal end toward the tail,
as well as the left and the right sides of the embryo. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node which contains a circular depression known as a primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak, forming a primitive groove. Once formed, cells of the epiblast migrate inwards towards the streak, detach from the epiblast, and slip beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate through the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are eventually completely replaced by a new proximal cell layer, which is referred to as the definitive endoderm.
By day 16, the majority of the hypoblast has been replaced. The remaining cells of the epiblast is now referred to as the ectoderm and form the most exterior distal layer. Some of the invaginated at the blast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form a germ layer known as the mesoderm. Once the formation of the definitive endoderm and mesoderm are complete, epiblast cells no longer migrate towards the primitive streak. Throughout gastrulation, the ectoderm continues to form from the cranial to the caudal end of the embryo, establishing three distinct primary germ layers throughout the whole embryonic disc. The gastrulation process is finally complete. Allah has stated in Surah An-Najm, verse 45, وَأَنَّهُ خَلَقَ الزَّوْجَيْنِ الذَّكَرَ وَالْأُنثَى. In this prominent verse, Allah has stated that human was created in two types of gender, male and female. In science perspective, a zygote is formed when a secondary oocyte ovulate and fertilize with sperm. If the sperm carries Y chromosome fertilized, a baby boy may be born. But if it is the sperm carries X chromosome fertilized, a baby girl may be born. Therefore, the baby's gender depends on the sperm that later fertilized with oocyte to form a zygote.
Allah has stated in Surah Al-Mu'minun, chapter 23, verse 14. ثم خلقنا النطفة علقة فخلقنا العلقة مضغة فخلقنا المضغة عظاما فكسونا العظام لحما ثم أنشأناه خلقا آخر فتبارك الله أحسن الخالقين. صدق الله العظيم. The drop or nutfah has been interpreted as the sperm or spermatozoa, but a more meaningful interpretation would be the zygote, which divides to form a blastocyst, which is implanted in the uterus, a place of rest.
Second, then we made the drop into a leash-like structure. The word alaqah refers to a leash or bloodsucker. This is an appropriate description of the human embryo from days 7 to 24 when it clings to the endometrium of the uterus, in the same way that a leash clings to the skin. Just as the leash derives blood from the host, the human embryo derives blood from the decidua or pregnant endometrium.
Then, of that leech-like structure, we made a chewlum. The Arabic word mudghah means chew substance or chewlum. Toward the end of the fourth week, the human embryo looks somewhat like a chewlum of flesh.
Then we develop out of it another creature. This implies that the bones and muscles result in the formation of another creature. This may refer to the human-like embryo that forms by the end of the 8th week. At this stage, it has distinctive human characteristics and possesses the primordia of all the internal and external organs and parts. After the 8th week, the human embryo is called a fetus. This may be the new creature to which the verse refers.
Allah has stated in Surah Nuh 71 verse 14, وَقَدْ خَلَقَكُمْ أَطْوَارًا. Though He has created you in various stages. The verse above state about the human created in stage. Human is such a very complex living thing. From the science view, the formation of a baby takes abundance of stages. Therefore, gastrulation in human is actually a very important stage